it starts counting down here, and I think that's when it's
Oh,
recording.
yes.
Yeah,
And your
okay.
light's on up there.
And your light's on. Yeah.
Yeah. Right. The light was on though when I was on, but I wasn't recording.
Somewhere. I'll have to play. Yeah, it could have. Yeah,
It might have been just doing audio. I don't know. Okay.
Okay.
All
And that's
right.
okay if we have that, because I see you later today. We get some more footage. Of
Yeah.
that.
Okay, but it's recording now. For
Okay.
sure.
All
Okay.
right. Well, how about go ahead and tell us your name and a little bit about your background, what got you into science, um, anything, hobbies, personal life too, whatever you want to
Okay.
share.
Do you want me to look at you or the camera? Or
Um,
what makes it easier
you
for
can
you?
go back and forth,
Back
whatever
and
makes
forth?
you comfortable.
Okay.
What I'm going to do is I might, and the bulk of this. I mean, I'm not just going to put it on the blog site. I might splice clips.
Okay.
Favorite clips. So we just have it as a recording device, and
Oh,
I
all
might
right.
take just like 30-second clip or something. So you don't have to worry that if it's perfect. Do I look here or there?
All right, sounds
Yeah,
good.
yeah.
Okay, so my name's Jose. I was born in Santa Monica, California, and I grew up there for about 8 years. Both of my parents immigrated into the country, so I'm a first generation college student. I moved to Arizona when I was about 7 or 8 years old, so I had most of my education there. I did my undergraduate degree at the University of Arizona in biochemistry and molecular and cellular biology, so it was kind of a dual program that they had down there. And then I decided to just go straight into medicine, so I didn't take a gap year or anything. So I just had those nice 2 months off to watch Netflix and do nothing, and then started medical school. So as far as what got me into science, when I was so kind of taking it back a little bit, the main reason why I wanted to be a doctor is that when I was 5, I had a I very vaguely remember a time when I almost actually passed away. I had a very high fever. I was the first child in my family from my parents, so they didn't really know how to handle that situation. And my fever got out of control. I lost consciousness, and I just remember sitting on the couch watching TV, and the next thing I know, I was in a hospital room, tubes sticking out of me, and then I just remember my parents being next to me, the doctor being there asking me all these questions, and that was kind of my first introduction into the medical field. And it wasn't until later that my parents kind of told me what happened during that time when I passed out. And what they had explained to me was that when they arrived at the hospital, both of them were immigrants, so English was their second language. They couldn't really communicate what was going on to the nurses and the doctors there. And it wasn't until a doctor who spoke Spanish that was walking by understood what my dad was saying, that I had passed out, I was in the car, I wasn't responding, that he rushed out, he got me, and then they brought me to the hospital after that because the nurses and the doctors there wanted me to fill out some forms, get me registered before they went to go back me. They went to go back to get me because they just didn't really understand what the situation was. Hearing that story is really what inspired me to go into medicine because I am very passionate about serving underserved communities, working with in Arizona. I worked with the free mobile clinic over there where we gave free medical services to people who were underinsured or had no insurance. We also had clinics where we went to Mexico and we held a free clinic once a month with free medical services for the community there in Mexico. Agua Prieta was the name of the city. And even here in Iowa City, I'm a member of the mobile clinic board where we go to different areas in Iowa City and also provide free medical care to underserved communities. So that's kind of what got me into the medicine route and why I decided to go to medical school and pursue this degree. What got me into science and research was kind of a whirlwind as well. I never thought I would do research in any part of my career ever until my undergraduate degree. For part of my biochemistry program, I needed to do a senior thesis. And also for medical school, I mean, it always looks good if you have some research under your belt. So if I'm being completely honest, the main reason why I got into research was because I wanted to get into medical school. So just something off to tick off, get another box, try to get into medical school. Unfortunately or fortunately, I fell in love with it. I picked up a pipette, and honestly, the rest of it is history. I love the fact that we were exploring questions that really didn't have answers. I liked that, even when you had an answer, you just ended up with more questions and more routes that you could explore. And it really it it kind of calls upon a different part of your brain and your thinking that learning and even medicine, I think, doesn't really get to, which is this aspect of in medicine, we practice a lot of what we know. You know, right now I'm taking 2 semesters, I mean, 2 years of just like, this is all the stuff we need to know. This is how you're going to diagnose patients. You know, you do this, this, and this. And it's a very checklist kind of way, the way we sometimes go about diagnosing patients. Uh, Whereas in research, there is no checklist because we don't know. We're we're kind of on that bound of what we know, what we think we know, and the complete unknown, which is what really drove me towards doing the basic science research that I'm doing right
And going back to what you said earlier, did you ever have a point where language was a barrier and you could tell there was a patient or family members that were very upset and you got to step in and translate what was going on? Or has there ever been a really critical situation where you were able to help someone where language would have been a barrier?
Yes, so especially a lot, even with my family members, when I was young, I would always go with my grandma to her appointments to the doctor because she speaks absolutely no English.
Mm-hmm.
So I would go and help her with translation. And even in the mobile clinic in Arizona, there were times when the provider did speak some Spanish and they would communicate, but sometimes they would have to call me over to help and translate with kind of the more technical terms when they were trying to get into the nitty gritty, things like that.
Did you ever find out how high your fever was?
No,
Okay.
no, I never did. No, I just remember my parents were telling me that I had passed out, and then I was in the car, and then at some point during the car, I stopped breathing. Um, yeah, and then when I arrived at the hospital, I wasn't breathing. I didn't have a heart rate. And yeah, I know.
And they
So,
must have been just frantic.
oh
because
yeah,
to stop and fill out papers, they couldn't.
right.
Just say,
That
I
was
can't
like
breathe.
the right. They were trying to get across that I couldn't breathe. I wouldn't have a heart rate. But at that time, you especially with English being their second language, they couldn't get that across.
So in particular, you're working in Dr. Walrath's lab with the lemon mutations in Drosophila.
Mhm.
What attracted you to that type of research?
Yeah, so the thing that I found fascinating about lemmonopathies goes back to a biochemistry course I took in undergrad. So the course was all about DNA. So it was 4 months of all we covered was DNA, everything from the transcription translation of DNA to some of the most common diseases and pathologies and treatments of problems with DNA. And I remember that even when we were covering the very basic stuff, we would reach a point where we discover some protein and we'd spend a good hour talking about, oh, this is what it does, this is the problems, these are some of the diseases that are caused, and this is what we do about it. Well, there was one time where we talked about the laminin proteins and about the structure of the nucleus. And we spent a good hour, 2 hours talking about the different functions and all the things that it does. And then we very briefly talked about, oh, these are laminopathies, these are the diseases. And then the professor just ended by saying, and we don't really know what else. And we moved on. And it was one of the very few topics where we discussed that little about the diseases, the treatments, the pathology of it. So when I saw that Dr. Lori Walrath was doing research on it, it really grabbed my attention because it was something that it seemed very obvious to me that we know very little about.
Can you describe some of the experiments that you work on in the lab, and especially for a lay audience that's new to this? What are these mutations about, and how are you investigating them in the lab?
Yeah. So in the lab, we basically work with mutations that are in a protein that basically surrounds the nucleus. So, okay, let me back up. So the nucleus is essentially a small ball that's in every cell and that contains DNA. And one of the many proteins that are around the nucleus is what's called a lamin protein. And the lamin protein has many, many, many different functions. And when there are mutations in these lamin proteins, you get a variety of different diseases that can arise. Although researchers have had great success identifying some of these mutations, the actual reason as to why a certain mutation causes one disease versus another disease is still widely unknown. There's a lot of great hypotheses, and there's been a lot of great work done trying to kind of phase that out. But the actual mechanism of A leads to B, that leads to C, which leads to all these other things, that pathway isn't really mapped out. So what we try to do in our lab is to try to find the different changes that can happen in the cell, whether it's the organization of the cell, the actual position of the nuclei, the way the muscles seem to be functioning, how these different kind of big picture things are affected by this single mutation, and how that could possibly lead to the various diseases that we're seeing. So in order to investigate this, I run kind of a wide spectrum of experiments, but they can basically be categorized into two different categories. The first of them is muscle function. So these are experiments where we try to assess the function of muscles that have these mutations to see whether they're better or worse compared to normal muscles that don't have these mutations. And to do all these experiments, we looked specifically at the fly model, the Drosophila madagascar. So through a well-established process, we essentially do some sort of DNA targeting where we introduce these mutations into the flies, and then in the larval stage, we're able to run tests to measure how fast the larvae are crawling, how far they're crawling, in order to try to quantify whether or not the muscles are better or worse. And we also look at the metamorphosis between the larvae stage and the fly stage, which is when they're using a lot of these muscles to move different things around to kind of metamorphosize into a full adult fly. And we measure how well that metamorphosis occurs by measuring the death and the death and survival of the pupae to the full-grown adult fly compared to a normal fly or a normal non-mutated protein to see how exactly the mutations make that metamorphosis better or worse.
Have you ever found something that surprised you? Some startling data where you got something you completely did not expect?
Yes, that happened this summer. We were looking at a new protein. Well, not a new protein, but I was trying to look at a protein called P62. And this protein is basically the cell's garbage man. He goes around, he finds proteins that are misfolded, proteins that are accumulating, proteins that shouldn't be where they are, and basically takes them to the garbage truck, which is the lysosomes, to basically being torn apart and completely disassembled. So one thing that is very common to a lot of the mutations that we have studied is that there's a lot of stuff in the cytoplasm, which is essentially the space outside of the nucleus but inside of the cell that shouldn't be there. So for example, we'll often see nuclear pores that should be only around the nucleus that are floating around the cytoplasm. Sometimes we'll see the actual lamin protein, which again should be surrounding the nucleus, but is for some reason floating in the cytoplasm. So my study was trying to determine how well this P62 garbage man is binding to all the junk that is the lamin in the cytoplasm. And what I found is that it's not binding any of it. And that was very, very surprising because, you know, these proteins are so abundant that Lori and I just kind of, we hypothesized that, well, there's all these proteins that shouldn't be there. P62 is going to go in there, bind them, and start destroying them. Well, it turns out that they're not binding them. And now the question is why? And that's actually the investigative line that I'm currently doing. I'm trying to look upstream of the pathway and downstream of this P62 garbage man pathway to see where is there a block, can we fix the block, and does fixing the block change any of the muscle phenotypes that we're seeing?
And a question, do you ever encounter that maybe there's not only a lamin mutation, there possibly is a mutation in the coding sequence of P62 or upstream or downstream? It's an interaction of two different things. Do you sometimes find that?
It could totally, it's something we haven't explored yet,
Okay.
but that could totally also be, there's so many different kind of what we say is like genetic background. So stuff that's away from our main principal focus, which is alignment, that could be indirectly or directly affecting these. But we haven't looked specifically at these P62 because presumably they would be wild type, but there's definitely a possibility that there's some mutation in there that may not lead to dysfunction per se, but could be leading to a misinteraction with the laminacy. But that's something we haven't investigated.
Um, when we think in terms of the laminopathies, it's just such a wide range of diseases. We have everything from muscle weakness to rapid aging in children, um, to strictly cardiac diseases. How could there be such different diseases arising from the same gene and so much variability? Are these unanswered questions, or are there any answers yet?
Well, it's just it can get so complicated so fast, and that's what I love and hate about biology, is that, like you said, there are so many different diseases that can come from these mutations that are, from our point of view, pretty similar. Sometimes they're very close to each other. Sometimes they're hypothesized by computer models to have the same effect on the protein. But when this mutation happens in humans, as you said, there's a wide range of phenotypes that we see, or a wide range of diseases that we see. So we're starting. I think the scientific community is starting to make way to identify this. A few years ago, I think, our knowledge was restricted to mutations in the DNA. That, oh, if these two individuals have different mutations, there must be another mutation that's kind of influencing it. And that was kind of our understanding of how DNA works, because, you have DNA, which codes for a protein, or which codes for mRNA, which then gets translated to make a protein, and that's, 1, 2, 3. We're now starting to realize that there's so much more that can influence these genes, that I think now we have different ways of looking at these diseases to try to phase that out. One example that comes to mind is something called epigenetics. And epigenetics is essentially the cell's way of regulating gene expression without actually changing DNA. And there are several ways that this can be done, one of which is changing the way that the DNA is wrapped in the nucleus by phosphorylating or unphosphorylating proteins in histones. And histones are essentially like it's like a small ball of a protein that DNA is wrapped around in the nucleus. And by changing the phosphorylation state of this histone protein, we're finding that it has huge implications as to whether our genes are turned on, whether they're turned off, whether the protein is made faster, made slower, and that's something that a few years ago nobody even looked at. And now the fact that we're starting to learn that some of these epigenetic changes can be inherited, I think it's giving us a different way of looking at and trying to phase out these differences that we see.
And you don't have to go very personal in this question. You can be more subjective. But as a young person in the world starting out, not just with your medical career, but presumably your personal life and families, there's been such an explosive growth even in the last few months or a year or two with direct-to-consumer genetic testing, sometimes even for couples that are dating and test your DNA. How do you think people of your generation feel about that? Are they ready to embrace this technology? Are they a little afraid of it? Is it overwhelming? Because, even a generation or two ago, people just met and really didn't know each other's genetic profile, but now you can be searching these kind of things, even as a dating couple. Just in general, with your age group, how do you think people react to that?
Well, I think what I've seen so far is that there's a kind of two people to kind of, it's not much of a spectrum. I think there's those that fully embrace it. Hey, this is so cool. We need to do it. I want to learn more about it. And there's other people who are very much of, I don't want to do it because it's my information. What are they going to do with it? Are they going to steal it, kind of thing? I think those are the two main approaches that I see kind of interacting with my own personal group of friends. Personally, I think it's really cool. I'm definitely in that group. I think that we're learning so much about DNA and genetics that if we were able to look at the population as a whole, instead of very small subset of either volunteers or patients who allow us to do um, these kind of genome sequences, we may be able to see patterns that we would otherwise not have seen without having that large data information. With that being said, there are definitely some dangerous implications that can be played in terms of, I've heard, things of finding like genes that like determine whether you're going to be aggressive, or genes that are going to determine, if you're more or less likely to determine crime, and that's kind of the blurry line where it can get pretty scary and pretty dangerous very fast.
And just for people that may be curious, is there a database where people have, donated their DNA either linked through Ancestry or 23andMe, and there's a pool of data? I forgot what the name of it is, but do you use that sometimes? If you think you found a change in a sequence of interest, you compare it to a pool of people, but it's still not totally representative of the whole population. And is it somewhere like 53,000?
Yeah, I don't. I've heard of it. I haven't used it personally. I know there is a database. I don't even know if it's here in the U.S. Um, we last semester we had a lecture about it. There's some hospital network somewhere where they essentially tried doing that, where they had every patient come in, give their DNA sample, and then they did mass kind of like statistical analysis to try to determine or find patterns in different people. Uh, I don't know if there's a database where you could go in and compare sequences of a mass number of volunteers who have given their DNA sequences. I think there is. I think I've heard of it, but I've personally never used it before.
Okay. And can you tell us a little bit more about people new to science might think you work on fruit flies? Do people ever look at you like, don't you work on mice? Or what about rabbits? What about bigger organisms? Can you explain to someone who's has a cursory interest in this, but not a lot of knowledge, why are people working with fruit flies?
So the fruit fly is, I'm going to be honest, I was one of those people. When I first started working with Lori, I did not believe in fruit flies, and now they are my babies. They are my children, and yeah. So fruit flies are a beautiful model organism because, well, first of all, they share many of many of the proteins and many of the pathways that humans have, the fruit flies have as well. Not only that, but they're sometimes simpler in the fruit flies that they are in humans, so it's easier to manipulate. It's easier to find patterns, and not only that, but their life cycle is very, very short, and their upkeep is very, very short. So the reason why fruit flies are so beautiful is because, again, I could set up my genetic process today, and by next week, I will have either larvae that I can run experiments with, or I can have adult flies that I can run experiments with. And if I were to do that in another model organism like a mouse, not only do you have to keep them for longer because they have to mature, but you also have to pay for that upkeep, which can also be very expensive. So the way that I've come to embrace the fruit fly is that it is honestly a very crucial part of the long process that is human discovery and trying to understand some of these changes that we're seeing. Um, I always like to think that even though it doesn't always occur like this, sometimes you have to start with cells, looking at individual cells in a petri dish. Then you can move to a model organism like the fruit fly, which is a lot more complicated because you're no longer just looking at individual cells, but a whole model organism and all the different complexities that come involved with that. And then you go ahead and move to maybe even a more complex model, like a mouse model, that is a little closer to humans, but also harder to take care of and more expensive. And then you slowly move your chain up until you can actually start looking at human tissue, human muscle, and then human subjects to kind of follow your hypothesis all the way through. And if you were to skip any of these steps, I think not only would it be detrimental to the research, but it would also be detrimental to your work in terms of, the time you put into it, the financial situation that needed to take care of all these organisms and all of these experiments.
And in your lab, I know you have interesting names for a couple of your cell lines. You've got a Jill-like fruit fly and a Priscilla-like fruit fly. What were some of the startling, surprising things that happened when you created those models? Lori had said a Priscilla-like fly was actually weak, and you were expecting it to be super strong.
Yeah.
So what happened there?
Yeah, it's fascinating because, sometimes like with Priscilla, you see that we expect certain phenotypes in the muscles to be a certain way simply because of some of the background information we have of patients that have those mutations. And when you model that in the fruit fly, sometimes they match up and sometimes they don't. And both of them, I think, are fabulous and great, because if they do match up, okay, awesome. Now we have a working sample that we can work with and try to determine what's going on and how is it going on and what can we do about it. Whereas where it doesn't match up, well, why? What is it that in a human this is detrimental, but when you model that in a fly, all of a sudden we don't see the phenotype that we would expect. So I think in both, we don't really know so far as to why some of those changes occur, but they open new avenues to investigation, which is very interesting.
So you haven't seen muscle overdevelopment or overstrength in your fly models?
No, not yet.
Which we do sometimes in the human.
Sometimes we do, yeah.
All right. I think we're closing on maybe the next interview, but do you want to close with just anything you'd like to say? Or what would you want people to know? Oh, I know. Here's what I was thinking about before. How is working in a lab in research going to affect your medical career in the future? Because I assume you want to practice as a physician. So what will you take with you? How will it affect how you interact with patients?
That's a pretty good question. Let me see. So I think, especially after you've practiced medicine for a little bit, I think it's easy to start pigeonholing patients a little bit. Maybe you have a patient who comes in who has pain when they go to the bathroom, who, describes this pain a certain way, and maybe 2-3 minutes into the encounter, because you've been practicing medicine for maybe 10-15 years, you know what's wrong, you know what you're going to do, you know what the next steps are. And it's very easy to, I think, get caught up into this algorithm that is medicine, which is great because it means that you have a lot of experience, you know what you're doing, and you're able to treat a patient, accurately and perfectly, and everything will be fine. But I think sometimes there could be the risk of losing that one-on-one connection, that realizing that the person you're sitting across with is another human being with their own personality, their own lifestyles, they're their own person. I think the thing research has taught me is that everything is super complicated, no matter how simple you may think it is, no matter if you think you can explain something, you can explain something up to the point to where you go, oh, nobody knows. And I think that working with these flies, working in my previous research lab, has really kind of instilled in me the mentality that, patients are very unique. And even though you may have two patients that have the same exact disease, that have a similar prognosis, that have a similar treatment, at the end of the day, they're their own individual human being, and they deserve to be treated as so, and not just kind of, oh, you're this kind of patient, and this is what we're going to do, and da 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 da. And I think that's something research has really opened my eyes into, that I thought I knew before, but now it's definitely instilled in me for sure.
Thank you so much.
Yeah.